Welcome you all to this cryo-EM course. Today we are going to start image reconstruction. Before going to image reconstruction or image processing, we want to go through the entire pipeline. What are the most important steps for image reconstruction? Previous class we finished about the sample preparation. I showed you the cryo-EM data collection for single particle analysis as well as for tomography also, how we can prepare sample for the tomography, sample preparation for thick sample. So, specifically for cell, tissues and mostly focused on the tomography. Now, why we are interested about single particle cryo-EM? There are couple of reason that we do not so if I just tell you that we do not need to get the crystal of the protein sample or DNA protein, RNA protein sample. So, any large protein molecules we can use for imaging. So, mostly if we just consider if protein size is above 100 kilodalton, so that will be the this is the sample molecular weight if I just say. So, if it is more than 100 kilodalton, it will be easier for us to go for single particle cryo-EM. And for the tomography, if you just consider that for small protein molecules you cannot do tomography. So, basically tomography we are using for large sample, that is basically for cell, tissues, for that we are using cryo-electron tomography. One more important part is that, so when we are talking that sample can be partially homogeneous for 3D and 2D reconstruction. Second one is that physiological condition it is possible because we do not want to involve with crystallization process. So, that is the reason we can use very moderate concentration of the protein sample. As I mentioned previously, for the cryo-EM, if we are just using the protein sample, generally 1.5 to 2 mg is protein sample is good enough. So, here so per ml is good enough for cryo-EM, where for the negative staining for 0.1 to 0.2 mg per ml sample is good. So, whenever we are trying to prepare our cryo sample, most of the time we have the concentrated sample. So, even if you have to concentrate the sample, first you concentrate the sample, make it 1.5 to 2 mg and then do the negative staining temp. So, even due to during the concentrating your sample, if there is some aggregation, you can figure it out from your negative stain. First you concentrate your sample, get that 1.5 to 2 mg per ml, then you dilute this sample maybe 10x or 20x dilution or 15x dilution and with the same buffer, and then check the negative stain. If sample is looking good, you can go for the next one. So, always remember that in this particular case we are using NaCl or KCl or very small amount salt concentration, and salt concentration will be 50 to 150 millimolar salt concentration, not very high, and then HIPS or TRIPS buffer. So, it is not like that we are using a huge amount salt concentration to get the crystal. So, in a physiological solution or normal buffer, whatever we are using, that 25 millimolar HIPS or TRIS and then 50 to 150 millimolar NaCl or KCl, we have the protein, we are feeding the sample and we are getting our resolving our structure. So, we can consider that samples is staying in a physiological condition. And as I mentioned previously also, for NMR or X-ray, we need a huge amount protein. Most of the time we are talking about 8 or 10 or even more mg per ml protein concentration, where you can see comparatively very less amount protein, comparatively for cryo-EM you can see very less amount protein we can use it. So, nowadays many steps are very rapid and automated, and we can, when you are using the cryo-electron tomography, we can do things in vivo condition. So, here, so take home message, if I ask you this question, so what is the advantages of the single particle analysis or cryo-electron tomography? So, the first point it will come, does not require crystallization, we do not have the crystal, we do not need X-ray crystallography at this moment. So, we do not need the crystallization trial and other things. So, the second point it will be, sample can be partially inhomogeneous, we can manage during the 2D and 3D classification, but yeah, definitely it will better if we have a homogeneous population. So, something we are expecting that if there is a little bit inhomogeneous, means partially inhomogeneous, we can manage. So, we are expecting 80 to 90% proper pure protein sample. Then third point it will be, sample will be in physiological condition all the time, and the small amount sample is required, that is the fourth point, and many steps are automated. So, things are really fast, and in vivo condition we can also try. Okay, that is one point. Then what is the limitation of doing all of these things? So, limitation it will be, if I just consider that, okay, what kind of limitation we are facing. Limitation. Okay, what limitation we have? First one, radiation. So, that will talk more, or we can call beam damage. Then second point is that image alignment. So, precision in the image alignment. Precision of precision of image alignment, what is that mean? So, whenever, so first let me discuss little bit about the radiation damage. So, as I mentioned previously that we are using the electron beam, and lambda, if you just consider, it is a very small size, that is 3.88 picometer for 100 kb, or if you just consider for 200 kb it will be 2.74, and 300 kb it will be 2.24, and when the wavelength will be shorter and shorter, it has lot of energy, and that energy can break the covalent bond. And most of the time when we are collecting our data, we are giving a long exposure. When you are giving a long exposure, it may break the covalent bond, it has that kind of energy. And then what will happen, we already discussed about the protein structure, that is the alpha helix or beta strand, that is the two secondary structure. But if you go to the alpha helix and beta strand, there will be a single amino acids, and they are connected, and all these single amino acids, they have side chains. So, lot of time because of this radiation damage, those side chain it will break. It may not break the peptide bond, but if you have a long duration very strong beam, it can damage the peptide bond also. Mostly that it will break the side chain, and it will create lots of noise. So, beam damage and radiation damage, it is coming because how much electron dose we are using. So, generally will suggest that, okay, you go for 50 electron per angstrom square, and the total exposure time you can use it some 8 seconds. And if you want to increase your electron dose, so sometime you can use 80 electron per angstrom square or 100 electron. So, in that time, so this is the electron dose, and this is our exposure time. this is our exposure time. In that case, if you are increasing the electron dose, previously you are using 40 electron, now you are using 80 electron, that time it will be better decrease that total exposure time. You can use it maybe 1 second or 2 second, something like that. So here generally for our purposes we reduce this electron dose more. So it is total something like that 5 electron per angstrom square and total exposure time. So this is the dose and total exposure time when we are using. So we are using 8 seconds or 10 seconds. So in those cases what will happen, total dose, so this is per second, and total dose, so here I am talking about the total dose. Okay, so total dose we are trying to keep it 40 or 50 or 80. So here the total dose, it will coming either 40 electron or 50 electrons, okay, per angstrom square. So same thing here also, that either it is 40 or 50 electrons per angstrom square. And here the, so take home message is that total electron dose will be 40 electron, or you can go for 80 electron also. That time you should not go for a long exposure time, you should reduce the exposure time 1 second, 1.2 second or 2 seconds, something like that. So you always have to consider how much total electron dose you want for your sample. Always remember that if you have a stronger electron dose and for the longer time, it will be radiation damage. Then precision in the image alignment. So that is another important point that there is a, when we are doing, we will discuss about that 2D classification, and we will discuss about this 2D classification. That time you will get that information how we are aligning our particle. So we are aligning our particle means there are multiple different orientation particles are there. We are segregating when same orientation particle together, and we are calculating the cross correlation between those particles. And our cryo-EM data is always very noisy. When we are calculating the cross correlation value between the two noisy particles, sometimes it is very difficult to get the perfect alignment, and that is giving us the wrong alignment and giving some average of all these wrong particles together. So the precision of the image alignment is the another problem. Then third problem is that number of particles. So number of particles, for single particle cryo-EM we need a huge amount number of particles. So generally, so that should satisfy all the Euler angle, and that is the reason you need a huge number of data set. So if you have a very small amount of data set, your and you are trying to calculate the average from them, and it will be not satisfying all the Euler angle and and that will give you a very low resolution structure. That is one of the reason whenever we are doing single particle analysis nowadays, your start point will be somewhere, if you just consider so something like this, and then probably from this data set, single molecule particle, from this data set probably only or next particle, so either particular this number 1 lakhs or 2 lakhs particle will give you the final 3D reconstruction. The fourth problem, it is our sample is staying in the physiological condition, it is in a normal buffer. When protein sample is in a normal buffer, we are not using a very high salt concentration or other component to get the crystal. It will be in a physiological condition and it has lots of motion, and immediately when we are freezing, whatever conformation we have in the protein structure, everything will be preserved there. So then not only that different orientation, that conformational heterogeneity is the another problem. So conformational heterogeneity Conformational heterogeneity is one more problem we can face. Now there is a fifth problem limitation I can tell, that it is preferred orientation. So what is that preferred orientation means? So lot of time when proteins are staying in the solution, lot of time they are just only one particular view it will present. So then you will not get all the side of that particular protein. If you don't get all the side of the particular protein, that is called the preferred orientation. If I just give you one example of the preferred orientation, you just consider that you have a sample something like this one. So if it is your 3D view, what do you expect? You will expect, if you look this object from top, you should see the circle. If you look at from the side, you can see this kind of thing. Now if you just look at this one little bit in a tilt view, what you will see? You will see something like that. If you look at from this angle, you will see something like that. So when we want to do a 3D reconstruction of this particular thing, we want all this view. So we want all this view and we want to back project them, and that will satisfy all the angle, all the Euler angle, and then we will get this 3D structure. Now lot of time protein, when it is staying in the solution, it has some particular nature and it just stay in one particular form. So it just stay in one particular form means what kind of particular form? It may just oriented like this. Whatever image you will collect, you will get only this circular view, means you are looking at this sample from the top. It is, so I already discussed how samples are staying in a ice. If this is your ice, so sample will only oriented like this. And then when electron beam will pass through this, you will see a 2D image where only this circular top view you can see it. So when you have this circular top view, that is called the preferred orientation. Means we don't have all the view, we don't have this side view. So that is, I told it, this is from the top view, right? This is from the side, this is partially side view. So here for 3D reconstruction we need all these views completely to get this 3-dimensional structure. So if in our images, if you cannot get all these view, that is called the preferred orientation. If there is a preferred orientation, what will happen? You will not get the high resolution structure. When you will try to resolve the structure, you can get the top view properly, you can see the top view properly. But what about this particular view? When you have this and you are getting the top view only, so this particular part you may resolve, or you can resolve the bottom part also, because you cannot distinguish from this one which one is top and which one is bottom. But what about the side views? So side view will be completely missing. So then what kind of reconstruction you will get it? You will get some kind of link and some kind of link, but this middle part will be completely missing because we don't have any information about this central part. So that is one of the major limitation when we are doing single particle cryo-EM. Okay. Third one, so now another important part, it is very much. So if I just consider that we already discussed about the 5 limitation, the 6th one, it is very much time consuming. Why it is time consuming? Because it is, we don't need to get the crystal, but there is a lot of screenings are required. What kind of screening? Because if you remember when I first talking about that, you have to do all sorts of biochemical experiment, when you have to do the cloning So all the molecular biology technique you have to follow. You have to do the cloning, you have to put the plasmid in, and then you have to go for a protein purification. Protein purification. When you have the protein purification, you take this protein and do the negative staining TEM. When you will do the negative staining TEM, lot of time we can realize that okay, my sample is not good, or it is aggregated, or it is falling apart, or this whatever protein sample I am looking for I am not getting. So on those cases you have to go back here and you have to start your molecular biology and biochemistry again. So before going for any imaging, it will be better to perform all sorts of biochemical experiment like SDS page gel to confirm your molecular weight, and then you should do the Western blot to confirm that is your protein. Sometime from SDS page gel will take that protein sample, we digest with trypsin and give it for mass spec to confirm that this is it is matching with the protein sequence. And so mass spec, then your protein size you measure by ACC, you can do secmols, you can do your CD, you can do rest of the other experiment like Western blot or any other confirmatory test. Even if your protein sample is active, whatever the assay you want to do it, and then go for the protein, then go for your negative staining. If everything is working fine, if negative staining you can calculate the concentration, if you have enough concentration, then you go for a cryo-EM. Lot of time during cryo-EM you can see the sample aggregation. So you can see the sample aggregation, you can see there is some other problems, proteins is not staying in the hole, or there is a preferred orientation, or there is some other problem. So lot of time you have to optimize the sample preparation in this particular stage, that I have a protein sample looks good, but there is a problem in our grid preparation or sample preparation or glow discharge. That time you have to play around lot here to optimize the sample. If everything is coming perfect, then you can go for structural calculation. That is the reason this is called a very time consuming process. It is not very straightforward. And seventh one, definitely the microscope itself, it is a very expensive. That is one of the problem that we all have to keep it in mind, that it is a all the consumables, whatever grid, then seeding, C-clipping, whatever you are using, itself microscope cost, maintenance, everything is very expensive. So always better to you have a very good purified sample and you target for cryo-EM, not before that. Okay, so what is the basic steps for our single particle cryo-EM? Let's discuss little bit about that, the basic steps what we want to do. So first step is grid preparation. So grid preparation, what do you have? So this is the most important step. So this is our most important step. And why the grid preparation? So there are lots of variation is there. You can just use 0.6 micrometer hole size or 1.2, 1.3 micrometer hole size, 2 by 2, 2 by 2 micrometer hole size. Those are the variable. There are many other types of grids are also possible. Sometime most of the things those are copper grid, sometime people are using gold grid, and then carbon coated grid on top of this. So there are this type of variability there. Then glow discharge. So glow discharge unit, so it based on your glow, so that plasma, so it is similar like plasma cleaning or glow discharge unit. That is, we already discussed about that, what is the milliamps you want to use it, how long you want to use it. So there are different types of grids are possible. One is called quantifoil one is called C flat. So most of the time if you just consider that quantifoil and C flat and use the similar kind of glow discharge parameter, it is going to it is not going to work. So generally for quantifoil, if you want to do cryo-EM single particle, generally we use 70 seconds to 90 seconds glow discharge and 20 milliamps. Okay, so 20 milliamps we are using 70 to 90 seconds. On the other hand, C flat, if you just use the same parameter and same parameter here, it is going to break the entire C flat grid. For the C flat grid, generally we are using a very small time frame, something 10 seconds or 20 seconds like that. So grid to grid we have to change. So that is one of the reason grid preparation is very important part, where what types of grid we are using, what types of glow discharge we are using, how long we are glow discharging. Then last step it is coming that what is the in vitro blot, when we are freezing our sample, what is the blot parameters. So blot parameters means blot force, then blot time and so on. So there are lots of other parameters are also there, what temperature we want to use, what humidity we want to use. So all these parameters we have to consider. So those are the part we have to be very careful about, what types of grids we are using, what type of glow discharge parameters we are using, what type of blotting parameters we are using. And if any particular sample, if these particular things are optimized, we don't want to change these things again. When the grid preparation is done, we have to be extra cautious to insert that grid inside the microscope, because everything should stay in the liquid nitrogen, otherwise ice contamination problem will be there. So handling should be as perfect as possible. After that, image collection. So again, image collection, there are a lot of things are important that we are going to discuss in the next class, that at what electron dose we want to use it. What electron dose we want to use it, what defocus value, we want to use it, and what should be the exposure time, what kind of ice do I want to image. So is it a thick ice, so thick or thin or moderate ice, and we have to adjust the eccentric height so it can calculate the defocus value properly, and we have to do the perfect alignment, so that we have to reduce the aberration as much as possible. So there should not be any astigmatism, there should not be any coma, so all the electron beam should be parallel before imaging, and then we are going to record. So image collection is the next important part. Whoever is helping for this data collection, they has to pay attention to all of these parameters. So starting from alignment, your grid, preparation, exposure time, dose, how the image acquisition is going on, then eccentric height, all the parameters, all are connected with your microscope. When that part is done, when your data collection is done, we got the data. So first we have to do the motion correction, that we are going to discuss more in next class. So next step will be that when we have a image, individual protein molecules are there. So we have to pull out all these individual protein molecules, that is called the particle picking. So we will perform this particle picking, and we can collect some pick particle 20,000, 30,000, 40,000, sometime 2 lakhs, 4 lakhs, 10 lakhs, and then from that we have to calculate a 2D class averages. So 2D class averages. So when this data collection done, and we are going for first all the images we will get it, we perform the motion correction because there will be a thermal drift or beam induced motion that we already discussed previously. When we are collecting the images, we are not collecting one single integrated image, it is a multiple frames are there. So we have to correct the motion, we have to align the images part by part, and then we have to get the integrated image. When you have the integrated image, that is called the motion correction. After motion correction, we have to perform the particle picking, and then we can go for 2D classification. So this is the part we are entering towards the 3D class, then we will take this one and go for a 3D classification. So this is the part, it is coming 3D reconstruction or 3D structure determination. So this is 3D calculation. In the next class, we will discuss more about that motion correction, particle picking, 2D classification and 3D calculation. And not only that, you always have to remember in between that one very important step it is there, that is called the CTF estimate. So CTF estimate, what is that CTF estimate? Why we are interested about the CTF estimate? So in between, after image collection, the CTF means contrast transfer function estimate. Next class we will discuss about this CTF estimate, and the CTF estimate is connected with our defocus value also. So how it is connected with the defocus value, that we are also going to discuss. So not in very details about the mathematical terms or something, it will just I will give you a rough idea, so you can connect those steps that okay, how what is happening when you are collecting the data, why we are suddenly talking about defocus value, and then the CTF estimate. So in next class we are going to discuss about that. Thank you all for your attention.